In the last video, we gave a broad overview of the processes of sensation and perception. We discussed how sensation and perception are certainly related, but also distinct. In the rest of the videos in this module, we'll talk more in depth about the details of how sensation and perception work for each of our different senses. In this video, we'll start with the process of vision. Each of our different senses is attuned to a different form of energy in the environment. In this case, our eyes, the primary sensory organ of our visual system, are attuned to light waves, which allow us to have the psychological experience of seeing the world around us. The process of vision begins when photons, particles of light, hit the eye, where they are transformed into electrical signals that can be understood by the neurons in your brain. First, light enters the eye through the pupil, a small opening that can change in size depending on ambient light levels. When light levels are low, such as at night, your pupil will dilate or expand to allow as much light to enter as possible. When light levels are high, such as in the middle of a sunny day, your pupil contracts. The pupil's size is controlled by muscles that are connected to the iris, which is the colored portion of your eye. After passing through the pupil, light then travels to the back of the eye, where it activates photoreceptor cells, which are specialized nerve cells that are designed to detect light. When light photons hit a photoreceptor cell, they can start an action potential, which then starts the process of transduction, converting that light energy into electrical energy that can be understood by your brain. Photoreceptor cells fall into two different general categories, rods and cones. Rod and cone cells are so named because of their different shapes. Cone cells are very sensitive to detail and are directly involved in our ability to perceive color, which we'll talk about more in a minute. Cones work best in bright light conditions, which is why it's hard to see the color of objects in dim light. Instead, everything just tends to look kind of gray. Rods, on the other hand, can't detect color, and they don't provide the same kind of detail as cone cells do. However, they are specialized to allow us to still see, even in low light conditions. Once enough rods and cones have been stimulated with photons, this begins the process of sending this visual information, via the electrical activation of neurons, to your brain. Specifically, information from rods and cones is first sent to the optic nerve, which carries information from the retina to the rest of the brain. At a point called the optic chiasm, the optic nerve diverges into two pathways. Remember our principle of contralateralization from Module 3.1? The same is true here. Information from your right visual field is sent to the left hemisphere of your brain, and vice versa. Once inside the brain, visual information is sent via a number of different structures, eventually making its way to the occipital lobe for further processing. So far, we've primarily discussed the sensation side of vision. That is, we've talked about how nerve cells in your eye transduce light energy into electrical energy that can be understood by your brain. But what about visual perception? That is, how does your brain actually construct your visual reality? It turns out that's more complicated and involves some top-down processes as well. For example, look at the following image. What do you see? There are actually two possible interpretations of this image. A young woman wearing a necklace with her face turned away, and an old woman with a large nose and her mouth slightly open. Don't worry, what you see has nothing to do with your personality. This is simply known as an ambiguous image, an image that has multiple competing interpretations. Here, the actual stimuli, the light hitting the photoreceptors in your eyes, is always exactly the same. But how your brain makes sense of that image can shift. And in fact, once you see both the old and young woman, your brain can effortlessly switch back and forth between either of these two interpretations. The top-down nature of visual perception can also lead us to make some predictable errors as well. For example, let's go back to the Mueller-Lyer illusion that we talked about in the very first video. Remember that although these two lines are in fact the exact same length, your brain perceives the bottom one to be longer. Why? Well, there's still some debate here, but one possible explanation for this is because of the way that your brain has been trained to perceive depth cues, especially in rectangular environments. And remember that individuals from indigenous cultures who were less exposed to perfectly right-angled rooms are also less susceptible to this illusion. In right-angled rooms, the angles in configuration typically corresponds to objects that are closer, whereas the angles out configuration typically corresponds to objects that are further away. Additionally, objects that are further away cast smaller images onto your retina, even though they're not actually smaller. Knowing this, your brain makes a correction for how it interprets the length of the two lines, and normally this correction would be appropriate, although in this particular case it leads to an error. Finally, let's talk about one specific application of visual perception, how we perceive color. There are two main theories of color perception, trichromatic theory and opponent process theory, and both have turned out to be correct. To understand how color perception works, though, we first need to remember that light within the visual spectrum is made up of different wavelengths. Light can have shorter and longer wavelengths than these, it's just that we can't detect them. The color we see as purple is made up of the shortest wavelengths that we can perceive, around 400 nanometers, whereas the light that we see as red consists of the longest wavelengths that we can perceive, around 700 nanometers. It turns out that we have different types of cone cells that are sensitive to different wavelengths of light. That is, cone cells will only fire if they are stimulated by light waves in the specific range that they're sensitive to. Specifically, we have three different types of cone cells. One type of cone cell that fires at shorter wavelengths in the blue range, another type of cone cell that fires at middle wavelengths in the green range, and a third type that fires at longer wavelengths in the red range. This was the idea behind trichromatic theory, that we have three different kinds of light receptor cells, each sensitive to a different part of the visual spectrum. It turns out that just these three colors, red, green, and blue, are enough because they can be combined to make any other color in the visual spectrum, as you can see here in this graphic. For example, red and green can be combined to make yellow, red and blue can be combined to make purple, etc. Even though trichromatic theory has been shown to be generally correct, it's also incomplete, and that's where opponent process theory comes in. Opponent process theory proposes that color is actually coded via three opponent channels: red-green, blue-yellow, and black-white. Red-green receptors can only send messages about one color at a time, so whichever color is stronger is perceived and the other is suppressed. Your brain then uses this information to compute a comparison of the difference between the two. One interesting effect of this is what's called an afterimage, the phenomenon whereby your eyes continue to perceive light from an object even after the stimulus is removed. Let's try this out. First, stare at this green and yellow flag for 30 to 60 seconds. Then, either close your eyes or stare immediately at a blank piece of white paper. I'll give you a minute to pause this video and try it if you'd like. What did you see? You should see an afterimage of the flag. However, the flag should appear blue and red, not green and yellow. Why did this happen? This is thought to be the result of opponent process. After staring at the flag, your green receptors became tired from all that continuous firing, while the opponent color, in this case red, was continually being suppressed. The result was that when you close your eyes, the color that was being suppressed becomes dominant, causing you to see a negative afterimage, or an afterimage consisting of the opposite colors. Some individuals' perception of color is also affected by a condition called color blindness. This condition occurs in around 7 to 10% of men and less than 1% of women. Color blindness is usually the result of a genetic mutation on the X chromosome that is thought to affect the functioning of one or more types of cone cells, although color blindness can also be the result of damage or injuries to the eye later in life. Although there are several different kinds of color blindness, the most common is red-green color blindness. Individuals who are red-green colorblind have difficulty distinguishing between red and green hues. For a video that explains color blindness in more detail, you can check out the supplemental video on Canvas. In this module, we covered the process of visual perception. For the exam, you should be able to describe how energy from light is transduced into neural signals that the brain can understand, describe the difference between rods and cones, discuss how top-down processing can lead to errors in visual perception, and describe how color perception works using trichromatic theory and opponent process theory.